Welcome. Today we're going to be looking at the cell cycle. It is something that all cells go through. As you go through, you're going to have this handout here that you'll be kind of answering as we go through the reading. Feel free to pause the video if you'd like, or answer at the end as we kind of make our way through this. Feel free to write all over your handout if you'd like to annotate as we go through. That may be helpful for your own purposes later on. So let's start at the top here. We're looking at the cell cycle. The cell cycle can be thought of as the life cycle of a cell. Like the life cycle of an animal or plant, an organism is born, has different phases of their life, and in terms of humans, the phases would be you start as a baby, you become a toddler, then you become a child, then an adolescent, a teenager, young adult, and then an old fogey. Each where they do different things and function in different ways. So when we're babies, we do different things and we function differently than we do as, say, teenagers. Of course, this is followed by death. Death is kind of that happens to all living things. So that is like the end of the life cycle. And cells, they experience death as well. Cells do this too. They start with a birth phase and they end with a death phase. Each cycle goes through a sequence of 4 stages during their life cycle. The 4 stages are gap 1, or G1, synthesis, or S, gap 2, also known as G2, and mitosis, known as M. These stages will be outlined below. But first, use the image of the cell cycle below to fill in the information on your associated worksheet to give a visual representation of the cell cycle. So as we scroll down here, you'll see this and you'll be able to fill this in on your handout. You might want to pause the video, take a moment, get that filled in, and if anything's confusing, let me know. The image above is laid out like a pie graph, which is a good representation of showing which stages of the cell cycle tend to spend the most time in. As you can see, the largest chunk of the pie above is the gap 1 or G1 stage, which illustrates this. So when we look right here, the whole pie would represent 100%. This is representing, well, if this is 50% and this is 25%, it looks like that would be what, close to like 40%. So like 40% of a cell's life is spent in the gap 1 stage. These stages of the cell cycle get their names from early studies of cell division. Scientists' observations were limited by the microscopes of the time. When a cell was not actively dividing, or reproducing, so to speak, they could not see activity in it. So they had these microscopes, but they weren't very powerful. They could see cells, but they couldn't see some of the fine, intricate details inside the cells because they were so small. However, when a cell was going through cell division, mitosis, they would actually be able to see stuff going on. As a result, they originally divided the cell cycle into 2 stages, interphase. Interphase was when they were looking at the microscope, the cell appeared to be at rest, not doing anything. And then the second stage, mitosis, that was when they saw something happening, namely cell division. Improved techniques and tools, and tools are a form of technology. We got to understand that technology is not just cell phones and internet and electricity. Technology is any advancement, and tools kind of helped advance our civilization. So tools or technology allowed scientists to detect or see copying of DNA. So they were actually able to see DNA being copied, which is known as DNA synthesis. To synthesize means to build, and we're building DNA. And they changed their description of the cell cycle to include the synthesis stage. Since they still could not see anything happening during other parts of interphase, scientists named the periods between mitosis and synthesis gap 1 and gap 2. Eventually, scientists learned that during interphase, cells carry out their normal functions and undergo critical growth in preparation for cell division. As we can see in the image above, interphase includes G1, S, and the G2 stages of the cell cycle. So when we look at this picture, we can see that interphase starts here, and interphase ends right here, which is where mitosis begins. Now included in interphase, we can see that gap 1 is included in interphase, so is S, and so is G2. Looking at the different stages of the cell cycle, starting with gap 1. The first stage of the cell cycle, as seen in the image above, is gap 1, or G1. During G1, a cell carries out its normal functions. If it is a skeletal muscle cell, it contracts to move the body. If it is an adrenal cell, it secretes hormones such as adrenaline. If it is an intestinal cell, it absorbs nutrients. During G1, cells also grow in size and organelles, such as the mitochondria. They increase in number. We spoke of the mitochondria when we were looking at cellular respiration. Recall the mitochondria provides energy to cells. When a cell is entering the first part of G1, right here, it's going to have only a few mitochondria, but by the time it gets to this part of the cell cycle in late G1, it's going to have more copies of the mitochondria organelle, as well as all the other organelles that happen to be in that cell. A cell spends most of its time in G1, although the length of this cell cycle, this stage, varies by cell type. For example, skin cells' life cycle is about 30 days. We talked about this in class. Whereas the life cycle of a stomach lasts 2 days. So in a skin cell, it takes 30 days to go from here all the way around to right here. But in a stomach cell, it takes 2 days to go from here to right there. During G1, the cell must pass a critical checkpoint before it can proceed to the synthesis stage. Checkpoints make sure that everything in the cell is ready to move on to the next stage, the S stage. Just as it would be dangerous for you to run a marathon if you had not slept or eaten for several days, it would also be dangerous for your cells to continue dividing if certain conditions were not met. So we kind of have checkpoints which proofread, kind of check out the cell, make sure that the cell is actually ready to proceed, because if it's not, we really kind of want it to stay in this phase that it's in. For instance, most animal cells need enough nutrition, adequate size, and relatively undamaged DNA to divide successfully. They also need specific signals from other cells telling them whether more cell division is needed, such as the case after a cut in the skin. So, in order to begin dividing, a cell needs to know when it's ready to divide. There would be indicators from other cells if there was damage saying, hey, we need more copies made. And also, once again, those checkpoints, they just make sure that a cell is actually ready to proceed before they actually do go to the next stage. The S stage is the synthesis stage. So the second part of the cell cycle is the S stage. Synthesis meaning the combining of parts to make a whole. During the S stage, the cell makes copy of its DNA. One set for each cell that is produced at the end of the cell cycle. We already know that one cell creates 2 cells. We also should hopefully know that both of those cells created need an exact copy of the DNA. So, by the end of the S stage, the cell has to have 2 complete sets of DNA, 1 copy for each of those 2 new cells. Gap 2. G2 is the third stage of the cell cycle. During G2, cells continue to carry out their normal functions, and additional growth occurs. Like G1, this stage includes a checkpoint. This checkpoint ensures that there are in fact 2 full sets of DNA and that the DNA is undamaged. It also checks to make sure that the cell is actually large enough for cell division to happen. So this checkpoint is kind of critical because this is the point where the cell checks to make sure that the DNA is complete, because one cell is going to create 2 cells. We need these 2 cells to have the exact same DNA. And if one of these 2 cells gets damaged DNA, then problems could certainly arise. Mitosis. Mitosis, the 4th stage in the cell cycle, includes 2 processes: mitosis and cytokinesis. Mitosis is the division of the cell nucleus and its contents. The contents would be the DNA, the organelles like the mitochondria. It kind of splits those up so that this cell gets some of those organelles, and so does this one. We don't want all of the organelles and all of the DNA to go to one cell, and the second cell gets nothing. During mitosis, the DNA, which is long and stringy, so kind of think of like a lot of yarn just kind of all tangled up. During mitosis, this long stringy DNA, it packs tightly together, and when this happens, DNA becomes chromosomes. So chromosomes and DNA are kind of the same thing. We can kind of think of DNA as yarn, and we can think of chromosomes as like a ball of yarn that's kind of been wound up tightly together. Recall that DNA has been copied during the S phase. So at this point, there are already 2 copies of each chromosome. 1 copy of each chromosome is then placed into the 2 different nuclei which are forming at this time. So this nucleus here contains the DNA. The nucleus kind of falls apart. DNA is released out into the cytoplasm or surrounding areas that are purple on here. Then we need to separate the chromosomes onto opposite sides of the cell, and then a new nucleus will form around each of those chromosomes during mitosis. Once the 2 nuclei are formed and each has a full set of DNA, so we have a full set of DNA here and a full set of DNA here, then cytokinesis occurs, and we see that happening on our screen. We can see cytokinesis happening. We can see this pinching here, which did not exist in this top image. Cytokinesis is when the cell actually divides into 2 different cells. The 2 cells that form as a result of cytokinesis are called daughter cells. They are genetically identical to the original cell. So if we take a look at the image below, we'll see an image of 1 original parent cell forming 2 new nuclei, then getting pinched in half during cytokinesis to form 2 daughter cells. So we see a parent cell. We see that we are starting to split chromosomes, which are really represented in these dark circles. These dark circles are moving to opposite sides of the cell, getting further and further apart. Then cytokinesis happens, where we start to split the cell in half, forming 2 cells. And these 2 cells, they are going to be exactly the same as the parent cell. So mitosis, it divides up the contents of the cell. Cytokinesis splits the cell.